I'm Allison Jonjak, the Cranberry Outreach Specialist with the University of Wisconsin-Madison Division of Extension. These videos are the on-demand recordings of UW Extension and the Wisconsin State Cranberry Growers Association's virtual Brown Bag Lunch series. These monthly sessions offer growers and researchers a way to share timely and relevant information. We hope you'll enjoy and continue to engage with us.
Hi, everyone. My name is Grant Holly. I'm the Executive Director of the Wisconsin State Cranberry Growers Association. We are very happy to be able to partner with UW Extension to bring you the research needed to be able to navigate our ever-changing environment. I'd like to thank all of our speakers today for their valuable time and leadership, the growers for joining us, and Allison for being the catalyst from which all of these sessions rely. Thank you so much.
I am recording. If you have a question, you're welcome to unmute yourself and ask a question, or you can put a question in the chat. I will be watching the chat and I can ask those questions to the presenter. If you'd like to ask a question anonymously, use the drop down and say ask a question to Allison Jonjak, and I will ask that question anonymously on your behalf. Today, I'll go ahead and jump into our agenda. We're going to flip the order here, and hopefully Dr. Shinya Ikeda will be able to go first and talk about an introduction to him, his work, his goals here. Then we'll have Dave Jones giving us some field updates, the Senior Ag Scientist at Ocean Spray. And then finally, I will give an overview of how to use the EPA Bulletins Live 2 website. But before we do that, I have been expecting to see Tom logging in, but I know Tom has a lot on his plate this week. And so in an absence of Tom at this particular moment, I will take the initiative to introduce Grant Holly to you. Dr. Grant Holly, who is the incoming Executive Director of the Wisconsin State Cranberry Growers Association. So give us your welcome.
Welcome. How's everybody doing? I'm super excited to be able to be here today. I'm at the research station. I've been here with a number of your leaders already, and I got to say that it's just one of the most healthy and happy communities I've ever been a part of. And thank you. This is day 5 of me being on the job, and I'm still learning as we go. But please reach out, and hopefully we'll all talk soon.
Yeah, thank you so much. We're extremely glad to be able to have. Grant it's been a fantastic 5 days. We're looking forward to the next many more.
It has. It's been a busy 5 days. It's yeah, cranberries are popping everywhere.
Awesome. So thank you, and look forward to seeing more of Grant in future. I assume we'll have introductions for the grower community and everything, but wanted to take the moment, since we were all here in one place, say hello. We'll let Dr. Ikeda share, and he is coming up upon not quite yet a full year as the food scientist, and so I'll let him introduce himself.
So hello everyone, I'm Shinya Ikeda, and I'm the food scientist in our group at USDA, and I have a lab in Babcock Hall, and this is Babcock Hall, and this building is the house for the Department of Food Science. We're just across the street from Moore Hall, where Dr. Zalapa and Dr. Mura are based in. Right now, I'm sitting behind this window right here, and I can see Moore Hall through this window. And my lab is located in the central area of the first floor of this building, and it was literally empty when I joined, and we are gradually setting it up. Here's the scope of my research, and my primary role as a food scientist is to elucidate how processing impacts product quality. And if you look at the process of SDC production, the key steps include the post-harvest frozen storage and thawing, and juice extraction, infusion, and drying. So I'm setting up my lab to process cranberries to make mock SDC products, sweetened-dried cranberry products in our lab, and study the impact of each processing step on product quality. Product quality also depends on fruit quality, obviously. And in previous studies, certain quality traits have already been identified as key quality traits. So I'm going to look into some of those key quality traits, including firmness and sugar, acid, and phytochemical compositions. And actually, I started my cranberry research from looking into fruit firmness because food texture has always been my core research interest. Also, here in the Department of Food Science, we have a food chemistry, biochemistry lab, which is my next door neighbor. And they have been actively studying phytochemicals in food, including cranberry phytochemicals and their health impacts. So currently, I'm working with them to see how phytochemical profiles depend on cranberry varieties and how processing impacts health benefits, actual health benefits of those phytochemicals. So I'm going to come back to this point later. And I also plan to work with Dr. Zalapa and Dr. Mueller to establish how genetic characteristics or field conditions impact fruit quality. And my primary role here would be to support them, their projects, by providing our analytical capabilities to determine chemical compositions and physical properties of cranberries. And that was the big picture of my research. And to be more specific, I'm currently working on 2 research goals. And this is the first one: identify the key compositional factors and structural factors that determine firmness. And last year, we published this article about the methods to evaluate cranberry texture. This project was led by Dr. Zalapa, and the main objective was to figure out what methods and test conditions are suitable to evaluate cranberry firmness. So we conducted various methods of texture analysis, including single compression, double compression, puncture, and shearing. And we were able to identify a few structural or textural traits that can be used as a good indicator of firmness. And one other key finding was that firmness is positively correlated with the fruit size, which means the larger the fruit is, the firmer it is. And the size is a structural factor. So this is a good starting point for my research. Then, well, when we test firmness using our fingers, we press the berry and feel the firmness, right? And when we test firmness using an instrument, we do essentially the same thing. We press a berry using a certain device and measure the force that the berry pushes back with. And as we press the berry more, the berry pushes back harder. Okay? So the raw data we get are forces measured at different levels of pressing. And from there, we calculate various textural traits, including firmness. But calculations like that always involve some kind of assumptions, particularly assumptions about the shape of a pressed berry. I mean, we know the exact force, which is measured, but we have to assume how the pressed berry deforms because it is not measured. And this is an example. This is an illustration of one of the models used in this publication. Here, sample is assumed to be round or spherical, which is obviously not the case for cranberries. Cranberries are more like an oval. But anyway, if we compress a sample like this, the sample should become a little bit wider as we compress it, right? But this model assumes that the width of the sample remains the same all the time during compression, which is obviously not the case. So currently, I'm working on to assess how useful this model is and also look at alternative models to find a hopefully better one. The first specific objective of the first goal is to seek correlations between textural traits such as firmness and structural characteristics. We recently purchased an X-ray micro CT scanner, which is a benchtop type, not like CT scanners for medical purposes. It's designed to test or scan relatively small things like fruits and vegetables, and that's why it's called a micro CT scanner. And these are CT images of a cranberry, and we can see 4 large pores in the middle, and we can also see numerous small pores within the flesh. And when I started cranberry research, I was impressed by the way how cranberries are harvested. And my first thought was that cranberries float on water because of these large pores in the middle. But soon I noticed in my lab that not only whole berries, but also slices float on water. Now I'm looking at these images and I know why even slices float on water. Yeah, that's because of these numerous small pores. And a good thing about our CT scanner is that we can derive quantitative information about the internal structure of berries without breaking berries, like the pore volumes, for example. And so currently we're working on to identify what structural characteristics are related to firmness. And another beauty of this micro CT scanner is that it has what is called a mechanical testing stage. So we use the mechanical testing stage to apply a certain force to a berry and compress it and then scan it. So it's like testing texture and structure at the same time. And I hope this approach will give us insights into the relationship between cranberry texture and structure. The second objective of goal 1 is this, to seek correlations between firmness and major metabolites, as well as correlations between firmness and cell wall components. Well, this idea came originally from Dr. Zalapa, and one of his students recently shared this paper with us where the author studied cell wall compositions and metabolite compositions for table grape berries with different firmness levels. And this approach makes sense because for foods derived from plants, in general, firmness is largely determined by 2 major factors: their turgor pressure and the mechanical strength of the cell wall. And the turgor pressure is related to the osmotic pressure within the cell. Then the osmotic pressure is determined by the number of dissolved molecules within the cell. So it makes sense that to determine the contents of metabolites in the cell. And as a matter of fact, these authors found that fruit firmness was positively correlated with increased concentrations of soluble sugars. And another interesting finding in this study is the relationship between firmness and cell wall components. They found high firmness was associated with an increase in glucose in the cell wall, and low firmness was associated with an increase in rhamnose in the cell wall. And glucose is a building block of cellulose, which is a major cell wall component that makes the cell wall rigid. So the more glucose, the firmer makes sense. Also, rhamnose is a component of pectin, which is another cell wall component. And pectin can cross-link with each other and make cell wall stronger, but rhamnose cannot be cross-linked, cannot be involved in cross-linking, meaning that more rhamnose, the less cross-linking. So overall, the more rhamnose, the softer, also makes sense, although the authors do not explicitly say so in this article. The second goal of my research is to elucidate how processing impacts the health beneficial effects of cranberry phytochemicals. And we're focusing on proanthocyanidins, or PACs, And cranberry packs are probably best known for its efficacy in preventing urinary tract infections. But they are also beneficial in preventing other diseases like cardiovascular diseases and infectious diseases. And packs are pretty complex molecules like these, and there are two types. Cranberries are rich in type A, which is rarely the case for most other plants. And the key function of cranberry packs is to prevent bacterial adhesion, and only type A has such an ability to prevent bacterial adhesion. And we are currently working on to determine pack and anthocyanin profiles of 19 varieties to identify varieties containing high, middle, and low levels of packs. And last year, we harvested berries at maturity at the research station. And we expect to complete the analysis sometime in this coming summer. Then we'll pick 2 varieties for each pack levels, high, low, middle, low. So 6 varieties total for the next objective, which is to dry cranberry slices in our lab and determine how that impacts the bioaccessibility of packs. Well, as I mentioned, I'm working on to develop a capability of producing mock SDC in our lab eventually, but as a first step, we'll be focusing on the drying step. We're going to look at how drying impacts the bioaccessibility of cranberry packs. And bioaccessibility means how much packs in a product like juice or SDC is actually available to function and provide health benefits. And packs in cranberries are either soluble or insoluble. Then cranberry juice contains soluble packs only. And the press cake or panache from juice extraction contains insoluble packs. And those insoluble packs are strongly bound to cellulose in the cell wall, so they're not available or accessible. So they cannot be utilized in our body to provide health benefits, even though they're there in the product. So bioaccessibility essentially means how much packs in a product is soluble and available. And drying at high temperatures is likely to degrade packs to some extent, but it is also possible that some insoluble packs may turn into soluble form due to high heat treatment. Well, in any case, it's important for us to understand the actual efficacy of a product, not just the content of active components. So, this is my introduction, my lab introduction and research goals. Thank you for your attention.
Thank you so much. Do we have any questions for Dr. Ikeda? I'm really impressed in an excellent way to see how much work you've been able to do in your, you've only been here not too very long yet. And so it's really exciting to see all of the work that you've already done. Very, very excellent. I especially liked the CT scans of berries and seeing all the so frequently we're told, oh yes, they float because of the pockets, but seeing those microscopic pockets as well is really neat for all of us. Any questions for Dr. Ikeda? Thank you so much for joining us. We'll continue to have questions on up to the end of the session. We had a question for Dr. Ikeda. Does Dr. Ikeda have preliminary results on levels of present and available packs in cranberry juice and SDCs, and how they compare with packs in non-processed cranberries? I did see that you replied to that question asker, but I'd love to have the answer out for everyone.
Yeah, so we haven't done anything yet for real products, but I'm aware of a report from Poland where they tested total polyphenol levels in raw cranberries SEC product they purchased at local market, and they found about 1/3 of total polyphenol remain in the product.
And that's compared to your 1/3 of what they were when they were fresh fruit, or 1/3 of when they
1/3 of the raw cranberry or fresh cranberry.
Okay, cool. That is a good result. And I'm sure we'll look forward to having additional to that as well. Other questions here? We've got, again, just a couple more minutes left in the hour. Any I'll go ahead and take questions, folks.